Welcome all to Genome, the GIS Seminar Series. Very proud to kick off this new series hosted by the Genome Institute of Singapore. Thank you all for joining. As you know, the GIS specializes in nucleic acids research, and so this seminar series will focus precisely on this topic, anything and anything related to genomics. And our very first speaker, we're proud to announce, is Dr. Sarah Tishkoff, who's the David and Lynn Silfen University Professor at the University of Pennsylvania and Director of the Penn Center for Global Genomics and Health Equity. Very timely, very important research, and Sarah's really in the vanguard of many new movements in genomics. Very broadly, she studies genomic and phenotypic variation in ethnically diverse Africans, and it's multidisciplinary. She does a lot of field work, as I've seen from her past talks and talking to her. Lab research, computational methods, all in service of inferring African population history, and how genetic variation can affect a wide range of practical issues. For example, why different people have different susceptibility to disease, how they metabolize drugs, how they adapt through evolution. And if, as you can imagine, in Africa, you have the greatest amount of genetic diversity. So actually the most important area of the world to do this kind of research in. So Sarah is a member of the National Academy of Sciences, recipient of the NIH Pioneer Award, David and Lucille Packard Career Award, Burroughs Wellcome Fund Career Award, many other awards. She's also on the National Academy of Sciences Board of Global Health, the SAB for the Packard Fellowships in Science and Engineering, editorial boards of many journals, including Cell, PLOS Genetics, Genome Research, and she's been funded by NIH, NSF, Chan Zuckerberg Institute, American Diabetes Association. Sarah, excited to hear your talk. Over to you.
Thank you so much. I'm really honored to be invited to give a talk to the Institute. I really wish I could be there in person, but on the other hand, I guess we have a much broader reach by doing it this way, and I still hope I'll get a chance to go there in person. So what I want to tell you today is about our research on evolution and adaptation in Africa and what the implications are both for health and disease. So I want to start by talking about what I think some of the key challenges are in human genomics research. The first is that we need to do a much better job characterizing both genomic and phenotypic variation in ethnically diverse populations. We want to understand the evolutionary processes that generate and maintain that variation, and to better understand how gene-gene, gene-protein, and gene-environment interactions contribute both to normal variation as well as disease risk. So the focus of my research, as you heard, is on Africa, and there are a number of reasons for that. But one of which is the fact that all modern humans originated in Africa. So these red dots represent the locations of fossils of anatomically modern humans, the oldest of which is dated to around 300,000 years ago. So after this origin in Africa in the past few 100,000 years, relatively small numbers of people migrated out of Africa and migrated across the globe, giving rise to populations around the world. When I say a small number, people have estimated it could have been as small as in the hundreds or low thousands. So you could imagine that this demographic history has really shaped the pattern of diversity that we see. And for this reason, we see higher levels of diversity in Africa relative to other parts of the world. Now, when modern humans left Africa, they ran into archaic populations like Neanderthals and Denisovans, and we now know that they interbred. And somewhere between around 2 to 6% of the genomes of people from outside of Africa can be traced back to these archaic populations. This is almost certainly happening in Africa as well. It's just that it's really hard to detect because we don't have any good archaic reference genomes. The other thing that motivates my research is to better understand risk factors for health disparities. So for example, hypertension is more common in people of African ancestry compared to other ethnic groups in the US. And type 2 diabetes is more common in people of Native American ancestry compared to other ethnic groups in the US. Now we know undoubtedly that there are major environmental, socio-demographic, dietary risk factors that are contributing to these health disparities. But it's thought that there may be underlying genetic risk factors as well. And yet, this was a perspective we wrote in 2019. In January of that year, we looked at the Genome-wide Association Studies database, and we're able to see that about 80% of the individuals included in GWAS at that time were of European ancestry. About 10% were of Asian ancestry, around 2% African ancestry, 1% Hispanic ancestry, and less than 1% everyone else. And if you look at other commonly used databases like Nomad or GTEx, you see again this very major European bias. So that's a real problem for a number of reasons. One is that it impedes our ability to fully understand the genetic risk factors for human disease, and it could actually exacerbate health inequalities. Our ability to translate genetic research into clinical practice or public health policy could be incomplete, or even worse, it could be mistaken. It could result in inaccurate assessment of pathogenic variants in clinical genomic studies. And one of the reasons for that is that one of the criteria for determining if a variant is pathogenic is how common is it in a population, with the idea being that if it's very common, then it's probably not pathogenic. But maybe this variant that's identified is not common in Europeans, but it's common in other populations. So that could actually change the assessment. And then lastly, attempts to use estimates of genetic risk from European-based studies and non-Europeans could result in inaccurate assessment of risk and lack of appropriate interventions. And I'm going to tell you a bit more about that. So I'm sure many of you now are familiar with polygenic risk scores, but for those of you who are not, when someone does a genome-wide association study, let's use as an example looking at heart disease, and these have been done predominantly in Europeans, tens of thousands, if not hundreds of thousands of people. And you can basically look at all the variants associated with risk for heart disease. And if you were to look at a patient, let's say you don't know what their risk is, and you look at the same genetic markers, and you basically are going to add up the effect of all of these different variants on risk for disease. So if I, for example, had a lot of the variants for high risk for heart disease, that would put my overall risk much higher than the general population. And you could imagine that this could be very important as a clinical tool. And in fact, it is actually being put into the clinic already. But a number of studies have shown, including this one by Alicia Martin and colleagues, that was based on a simulation, but there have been empirical data to show that when you calculate polygenic risk scores in Europeans, and then you try to apply these to patients from other ethnic groups, and you try to predict the same phenotype or risk for disease, it does really poorly. And it does particularly poorly in East Asian and African populations. So why is that? There are several possibilities. So one could be that actually it's exactly the same causal variant, or it could be the exact same causal gene or genes. But when we do a GWAS, we're looking for variants that are associated or tag the causal variant. Very rarely do we actually know what the causal variant is. And it just could be that the tag variant in Europeans is different than Asians and different than in Africans. Now, in theory, we should be able to address that if we can just find better tag SNPs in these other populations. But alternatively, there could be different causal variants. So maybe a different genetic architecture in a population due to different demographic history or adaptation to different environments. And if that's the case and there are different causal variants, you're never going to find them unless you look in ethnically diverse populations. So I've just discussed these first two causes of lack of replication of polygenic risk scores. And then also there could be other things like there could be different effect sizes in different populations due to differences in allele frequency, gene by gene, or gene by environment interactions. So because of this major bias towards Europeans, my research, as I said, is focused on Africa. I just want to take a moment to tell you about diversity in Africa. There are over 2,000 languages spoken in Africa, and those languages can be classified into language families based on similarities. So in blue is the distribution of people who speak Afro-Asiatic languages. These could include Cushitic or Semitic languages. In red is the distribution of people who speak Nilo-Saharan languages, mainly in Central and Eastern Africa. People who speak Nilo-Saharan languages are traditionally have a pastoralist lifestyle. And then the most widespread language family is shown in orange, Niger Kordofanian, also known as Niger Congo. And that originated in West Africa. The largest subfamily are called the Bantu languages, and they originated around the border of Cameroon and Nigeria. And people who spoke these languages had developed iron tool technologies that allowed them to cut down the forest and to do what's called slash and burn agriculture. So they were very effective at growing crops and sustaining really big population sizes. And so they spread across Africa to the east and the south and to the west and to the south, and they really shaped the pattern of variation we see in Africa today. And the last language family, as shown in green, is classified as Khoisan, and these are people who speak with clicks. I'm not able to do it myself, but they would include the San populations in Southern Africa and two groups in Tanzania called the Hadza and the Sandawe. So I just want to start by saying that this has been a team effort. We work closely with collaborators throughout Africa, and this is just showing some of our collaborators. And I thought I'd also show a bit of what it's like to do this research. We're mainly studying minority populations in Africa that are living more traditional lifestyles. So they tend to live in more remote areas, and that requires bringing a four-wheel drive vehicle, and we have to take all of our supplies with us. So here you get the outside perspective, and here you get the inside perspective. And this is in Tanzania. The individuals on the upper right and lower left are Hadza hunter-gatherers. I mentioned speak with clicks. And in the upper left is a Nilo-Saharan pastoralist. And here you could see at top, this is how we're doing some of our biometric measurements. And this is from Cameroon. These are a number of my colleagues from the University of Yaoundé Medical School and folks in my lab who have been assisting with this research. And one of the things that we're really, really careful about is making sure that this research is done in an ethical manner. And what that means is that we don't just get IRB approval at our universities. We have to go through a very long ethical review process in each country. We get research permits. We spend a lot of time going to the different villages, explaining what we're doing, translating into the local language in layman's terms, and talking about the risks and the benefits, if any, and so on. And after we have community consent, we then ultimately have to get individual consent. We also think it's really important to return results to participants. No, the Hadza don't read science, but we were able to summarize and to put this again in terms that they can understand and then translate. And we could also even show them some of the figures that I'm going to show you today in my talk. And I can tell you that people really, really appreciate this. And I think it's a way of benefit sharing. And then training and capacity building is really important. And I've had the honor of training a number of grad students and postdocs shown here, because the ultimate goal is that they should bring this experience back to Africa and should be able to lead research on the continent. So in these regions, we obtain blood, and from blood, you can get DNA and RNA. And when we bring liquid nitrogen, you can get frozen plasma. We also get fecal samples to look at the gut microbiome. We get very detailed information about ethnographic information and information about diet and any medical history they might have. And then we have to process these samples in regions without electricity. So what we've started doing in the lower left, sort of out of the frame here, but we started bringing a portable generator, and so we can just set up a lab in the bush. And then we measure phenotypic diversity in any way we can do it in a non-invasive manner. So this includes very detailed anthropometric measurements, cardiovascular, lung and blood phenotypes, metabolic function, and infectious disease status when we can. And then we're using what I call an integrative omics approach. At this point, we're starting to do high coverage whole genome sequencing. We're doing some proteomics from blood. We haven't yet done a lot of epigenetics looking at methylation patterns, but I'm quite interested in doing that in the future. We look at gene expression both from whole blood and also from single cells before and after immune challenges. And we try to integrate that with genetic data to find variants that are correlated or influencing gene expression. We've looked at the gut microbiome. We've looked at the blood metabolome. And then we look at how these interact with each other and how they're influenced by environmental factors and things like diet and pathogen exposure, and how those influence the traits that we're looking at, as well as disease risk. So the populations that we study, while they're very different from each other in different regions and they have very different environments, because they're in very rural areas, they tend to have a pretty homogeneous environment. So they tend to eat the same foods. They have the same sort of cultural practices. So one thing that we're able to do then is compare people, let's say, in a rural versus an urban environment who share the same genetic ancestry, and to try to distinguish the role of genetics and environment on variable traits. Or we could do the opposite. We could look at groups that have very different genetic ancestries, as shown by these different colors here, but live in the same environment or a similar environment and have different phenotypes. So for example, the Fulani pastoralists are known to have relative resistance to malaria for reasons that we don't understand compared to neighboring groups. And the San in Southern Africa have susceptibility to TB, tuberculosis, relative to other groups for reasons we don't understand. So I'm going to first tell you about our study that was done a while ago, back in 2009, but it remains one of the largest studies of genomic variation in ethnically diverse Africans. We looked at over 3,000 Africans from 121 ethnic groups shown here, mainly in places where we were doing field work, but you can see there's a lot of gaps. So there's still a lot of diversity in Africa that's not represented here. We then use computational approaches to infer the number of genetically defined ancestral or genetically differentiated ancestral populations. And those are represented by the different colors. And this plot is made up of a number of lines, and each line is an individual. And we can infer the ancestry for each individual. So here in blue are people who self-identify as European or Middle Eastern. Here we have people from India, from Pakistan, from Central Asia, from East Asia, from Oceania, from the Americas. And if we look here at Africa, you can just see by all the colors how much genetic diversity there is amongst populations in Africa compared to what we see across the entire globe. And this is perhaps even better demonstrated if we just focus on Africa, and we've pooled individuals by language or geographic region, and we're showing the proportion of different ancestries. And you can see by all the colors how much variation there is, and groups in Eastern Africa, Southern, Central, Western, and Northern are genetically differentiated. And we could also see, if you just look at the faces here, a lot of morphological differences, physiological differences, and this is due to the demographic history of Africa and adaptation to very different environments and diets. So I want to tell you about a study that was done by a postdoc of mine, Xiaohua Fan, who is now a professor at Fudan University. And this was done in collaboration with David Reich's group. We sequenced at high coverage genome sequence. We generated high coverage genome sequence from 94 individuals from 44 African populations. And if we construct a phylogenetic tree, at the end of each of these branches is a population, and if the populations are genetically similar to each other, they're going to cluster close to each other. So we could see that the populations with the oldest genetic lineages, the ones that are most basal at the root of all other lineages, are the San, followed by the so-called Pygmies, Central African hunter-gatherers. And then we see that populations cluster by geographic region, West Africa, and then East Africa. And if we were to add non-Africans, they'd be up here, basically a subset of the diversity that we see in Africa. We can also use computational approaches, coalescent modeling, to make inferences about changes in the effective population size going back in time. So the effective population size would be the number of, essentially number of breeding individuals. And we could see that in the past, let's say between 50,000 and 200,000 years ago, the San and the Pygmy hunter-gatherers had the largest effective population size, even though their census size is pretty small today. If we were to look at non-Africans, they'd be right around here, around 10,000. We can also make inferences about times of population divergence. So the oldest divergence is between the San and all other populations greater than 100,000 years ago. Some have estimated it could have been over 200,000 years ago. We see divergence between the San and some of the other hunter-gatherer groups around 80,000 to 90,000 years ago. And then even the other populations are very old, splitting somewhere between 20,000 to 40,000 years ago. So the point is that there's been a history of population substructure in Africa for a very long period of time and a very complex demographic history. So more recently, we've done high coverage whole genome sequencing. This is from 180 individuals. And just even with that relatively small number of people, we found millions of novel variants shown in red here compared to the databases. And many of these are predicted to be functional, either variants that alter protein structure or are in regulatory regions. I next want to tell you about the level of phenotypic variation we see in Africa for some of these traits. And this is work done by Matt Hansen in my lab, and he's been integrating this with genotype data from around 2,000 individuals from these populations. And these are groups that have very different diets. So hunter-gatherers have a diet heavy in vegetables, fruits, nuts, and tubers, and to a lesser extent meats, and pastoralists have a diet heavy in milk, blood, and meat, and farmers have a diet heavy in grain and maize. And we see a lot of variation. So if we look at height, and I believe this was just for women, and then he color-coded it by subsistence, we see that the shortest people are the so-called pygmies, and the tallest people tend to be the pastoralist groups. Whereas if we look at BMI, the pastoralists have the lowest BMI, so they're very tall and thin. And the food-producing groups tend to be on the higher end of the BMI. Similar pattern with waist circumference at the low end are the San populations. At the higher end tend to be the food-producing groups. And percent body fat, again, similar trend. The highest are the Herero, and you can actually see this is a photo of a Herero woman, and it is considered culturally preferable for women to be very heavy because they think that this indicates that they're able to have lots of children. So for that reason, we tend to see a much higher body weight. If we look at C-peptide and glucose, again, we can see not as striking differences, but you do tend to see differences based on diet and ancestry. So in fact, what Matt wanted to do is he then repeated this analysis where we infer individual ancestry, and he wanted to know how much of this variation is correlated with genetics and how much is correlated with environment and how much is attributed to ancestry. And so the way you can think about this, it's a heritability analysis. And when you see this dark bar, think of that as the genetic contribution to the trait. And if you see an open space here, that's the environmental contribution. And then the genetic contribution is broken down by that which is contributed by genetic ancestry versus variation within a population. And what we see is that skin color is most influenced by genetic variation, and also a number of the anthropometric measurements like height and knee length and arm length, and even cholesterol and grip strength. But a number of these are more influenced by environment, including blood pressure and sugar levels and some lipids and so on. More recently, we've been doing targeted lipidomic analysis with Chuck Barant. And we could see that when we look at a PCA based on levels of 700 lipids, that you could see some clustering based on subsistence patterns. So here are the pastoralists and here are the agriculturalists. And in fact, we can identify lipids that tend to be different in prevalence in different groups. So the agriculturalists, for example, have higher levels of free fatty acids, and the agriculturalists, oh, I'm sorry, the pastoralists have higher levels of di and triglycerides. And we've tried to tease this apart a little bit more. For example, if we compare these two groups, the Weto shown here and the Amhara, they have a pretty similar genetic ancestry. There's some small difference, but pretty similar. But the Weto are foragers and the Amhara are agriculturalists. And despite the genetic similarities, we see differences here. And here's a case where the Hamer and the Mersey, so here's Mersey and here's Hamer, have different genetic ancestry, but similar diets, and yet they're still showing variation. And if we look at the Hadza, some of whom are more traditional and some of whom are more settled lifestyle and different diets, we can't distinguish. So it's a little confusing. It's hard to disentangle what's the role of genetics and environment. So we've then, to try to disentangle this, we integrated this data with genomic data, and we identified hundreds of variants associated with these lipids. And not surprisingly, the lipids tend to be correlated with each other, and the genetic variants are often correlated according to metabolic pathways. But what was interesting, one of the things that could explain these observations we saw is if you just look at the relative level of triglycerides, this particular triglyceride, it's highest in the Dizzy pastoralists, it's lower actually in the Hadza and the Herero. But within each population, you could see that they're clearly influenced by the genotype here. So genetics is having an impact, but so is diet. So I think that's the take home, and we're still trying to disentangle those factors. Now for the last part of my talk, I'm going to talk about an example of adaptation to different environments. This shows you a number of examples. Many of you are familiar, I'm sure, with sickle cell disease being adaptive because people who are heterozygous are protected against malaria. Lactose tolerance is an example, but so is skin color. So in blue, I'm showing you the inferred melanin levels, and melanin is the pigment in skin that gives it a dark color. And we could see it's highly correlated with UV levels. And it's thought that this is an adaptive trait, that when populations migrated out of Africa into higher latitudes, that there would have been selection for lighter skin to facilitate production of vitamin D, which is synthesized through the skin. And there'd be selection for darker skin in places closer to the equator. But most studies were done in Europeans. So to study this in Africans, we use a spectrophotometer, and you shine light under the arm and you look at the wavelength of the reflection. And from that wavelength, you can infer the melanin levels. And what we see is a big range in Africa, with the most lightly pigmented people being the San hunter-gatherers from Southern Africa. And then the most darkly pigmented people are the Nilo-Saharan speaking groups that originated in South Sudan. So this variation is not due to admixture with non-Africans. This is naturally occurring variation. We then two individuals in my group, Nick Crawford and Matt Hansen, did a genome-wide association study of a relatively small number of people, 1,570 people. And even with that small number, we were able to find 8 independent associations at genome-wide significance levels in 4 regions of the genome. And I'm just going to step through those. So the strongest association was at SLC24A5. And this was actually the first gene ever identified to play a role in light skin color in Europeans. And there is a non-synonymous variant in this gene. And people who have the allele associated with light skin are shown here in the blue color. And that's at almost 100% frequency in Europeans. And it's also very common in Pakistan and India, not common in East Asia. But we see it quite common in East Africa, lesser extent in Southern Africa. So the question we had was, did this arise independently in Africa, or was it introduced by migration? So to address that question, we constructed haplotypes, a haplotype network. So a haplotype is looking at how different alleles are arranged along a short region of a chromosome. In this case, we're looking at about 70,000 base pairs. And each haplotype is indicated by a circle, and the size of the circle indicates the number of people who have the haplotype. And then the color indicates the relative frequency in different populations. And this is sort of like a phylogeny because this haplotype, for example, is very different from this one. This haplotype might differ by just a couple of mutation events, for example, from this. And what we could see is that non-Africans have the non-synonymous variant is on a common long haplotype consistent with what you might expect to see if there's recent positive selection on that variant. But we see that in East Africa, the variant's on the same identical haplotype background. So that indicates to us that this was likely introduced due to back migration into Africa from outside of Africa. And then it may have been under selection once it was introduced back into Africa. The second strongest association was at a gene which at the time didn't have a name. It was just called open reading frame, whatever. And then it got a name, when we were in the middle of the study based on homology to the major facilitator superfamily of transmembrane transporters. So this is called MFSD12. It's thought to facilitate transport of ions, amino acids, and peptides. And the only clue that we had that maybe this has something to do with skin color was a study looking at gene expression in the lightly pigmented and the darkly pigmented skin of people with vitiligo. And this was one of the most significantly different mRNA levels. And then our collaborator, Elena Owansia, found something very similar. She had independently been looking at this in her lab, and she found something similar. And she found that it's also expressed at very high levels in melanocytes relative to other types of skin cells. So we found two independent associations at MFSD12. One is within the MFSD12 gene, and we could see that variants associated with dark skin are found only in Africa and populations of recent African ancestry like Afro-Caribbeans and African Americans. And then if we look at the variant upstream, we showed that that's in an enhancer. And that we could see that the variant associated, I should say that's regulating expression of MFSD12. And we can see that the variant associated with light skin is common in Europe, East Asia, to a lesser extent in India and Pakistan. And it's common in East Africa, and it's very common in the sun. We then collaborated with Kevin Brown and colleagues at NIH and a grad student in my lab, Derek Kelly, to look at the transcriptome from 106 melanocytes, 15 of which were from individuals with high African ancestry. And Derek showed that high African ancestry was correlated with lower levels of MFSD12 gene expression, and that the variant associated with dark skin pigmentation is associated with lower levels of MFSD12. Then Marcia Beltram, a postdoc in the lab, used luciferase expression assays to show a very dramatic, that this was definitely an enhancer in this upstream region. And then our collaborators, Mickey Marks and Shannon Bauman at Children's Hospital, CHOP, were looking at mouse melanocytes. And what they did is they knocked out the mRNA for MFSD12 in the mouse. And when they knock it out, what they see is an increase in the melanin levels. So that's very consistent with what we were seeing, that there are higher levels in people who are more lightly pigmented. But what was odd is that they didn't see it co-localizing with other genes that are expressed in the melanocytes or melanosomes, the place where the melanin is produced. But instead they saw it expressed in lysosomes or some lysosome-like structure, which was surprising and indicating that it's working in a different way than had been previously understood. And then in collaboration with Bill Pavin and his group at NIH, they knocked out this gene using CRISPR-Cas9 and had a really dramatic impact on the phenotype. So we see the loss, this is an agouti mouse. So we see the loss of this sort of brown, reddish, yellow color. We see the underlying gray color of the mouse. And Stacy Loftus, who's part of that group, when they first saw this mouse, they said, you know, that looks really familiar. I think we've seen this mouse before. And it's a mouse that you can actually order from the Jackson Labs. Shown here, this is called the grizzled mouse, grizzled phenotype. This has been characterized since the 1960s, and they tried to map the mutation causing this phenotype in the 1990s, and they mapped it to around a 2 million base pair region syntenic to MFSD12. So Stacy sequenced the genome, and the only place where she found a difference was a 9 base pair in-frame deletion in exon 2 of MFSD12. So showing that that same gene is influencing that phenotype in the mouse. So I like to say this is a very rare example where we did a human GWAS to map a mouse mutation. So usually it's the opposite, done the opposite way, but we are very proud of that. So this is our other collaborators, Yoon Song and Ethan Jewett. And they constructed gene genealogy. So imagine if you just unwrap this, it's kind of like that phylogenetic tree that I was showing you earlier. And the lineages that have the light allele have this open circle. And these are the lineages that have the dark allele. And then the colors represent which populations they're in. So generally these purple and blue colors are non-African populations. And what we could see is that at this gene, the ancestral allele is the one associated with the light skin color, and that the derived allele is almost 1 million years old. So these variants have been segregating for a long time. Now, the third region was near DDB1, and that protein plays a very important role in repair of DNA in response to UV damage. People who have mutations in this gene or in the proteins that interact with this gene have a disease called xeroderma pigmentosum. People who have this disease can't go out into sunlight because they can't repair the DNA damage and they're prone to getting skin cancer. But what does that have to do with skin color in humans? We don't know, but interestingly, DDB1 is the gene that influences the color of tomatoes. So we're still working on that, trying to figure out how this influences pigmentation. Again, we found 2 independent associations, both upstream of DDB1, but we were able to show that these are in enhancer regions and show chromatin looping to DDB1. Now, I should note that this variant is part of a long haplotype block, so it's really hard to say what are the causal variants. This is just one possible candidate. In this case, high African ancestry is correlated with high levels of DDB1. So it's like the opposite pattern of what we saw for MFSD12. And again, the dark alleles are associated with higher levels of DDB1. We were able to show that these upstream regions are acting as strong enhancers. And then Xiaofen looked at signatures of natural selection, and he used something called a Tajima's D statistic. The most important thing to know about this is that it's based on the allele frequency distribution, but if it's a negative value, then that is consistent with recent positive selection, so that the variants rise rapidly to high frequency. If you see a positive value, that would be more consistent with balancing selection, like what you see for sickle cell, for example. And then we looked at the 1,000 genome samples as well as some of our own samples. And he saw a really striking signature of positive selection in the Chinese and in the Europeans, over hundreds of thousands of base pairs. And if you look at the level of diversity, the heterozygosity level, it's really striking. It's pretty much wiped out in Asia. I mean, there's almost no diversity in Asia for hundreds of thousands of base pairs. Not quite as striking in Europe, but very different than what we see in Africa. And if we look at the gene genealogy, or actually the haplotype network, this is over more than 200,000 base pairs. That's a really long haplotype. And we see this one really common haplotype in non-Africans. So that's very consistent with the strong positive selection. And if you look at the gene genealogy, you see this rapid coalescence at around 60,000 years ago of lineages outside of Africa. And that's interesting because that corresponds with the time of migration of modern humans out of Africa. So I think this is a very rare example of a near complete selective sweep when modern humans migrated out of Africa. The last region that we looked at, excuse me, is at OCA2 and HERC2, and that was well known to play a role in pigmentation, and there had been variants identified in HERC2 that are associated with light skin color and blue eye color in Europeans. But the variants that we found arose independently from the European variants. So these are different. And we show that these are in enhancer regions in HERC2 associated with expression of OCA2. And then there was this synonymous variant in exon 10. And the variant associated with light pigmentation is more common in Europe. We also see it in a bit in East Asia and South India, and then in the San population. And it was not associated with gene expression, but it was associated with alternative splicing. So the light allele, the allele associated with lighter skin color, is associated with a shorter transcript that's missing exon 10. It's missing the entire exon, but it's still an in-frame mutation. So there's still a protein produced, but the protein is missing the third transmembrane region. So you can imagine that's going to have a really strong impact on the function of that protein. And in this case, we saw evidence for balancing selection in the San population in that region. In the HERC2 region, we saw evidence more for positive selection in the Europeans. And here we see this very old time to most recent common ancestry over 2 million years. It's much older than what we see at most genes, consistent with the possibility of balancing selection, maintaining those two alleles. So in summary, in half of the cases, the ancestral allele was the one associated with light skin color, not dark skin color, and the age of the derived mutation is older than the origin of modern humans. So what does that tell us about the evolution of skin color? Well, this poor chimp lost its body hair, but you can see it has relatively lightly pigmented skin. I've heard from anthropologists this is not true of all chimps, but many do have relatively light skin. And it's been hypothesized that that would have been the ancestral state, and that when our ancestors migrated out of the forest and into the savanna, there would have been selection for the loss of body hair and increased number of sweat glands to regulate, to have better thermoregulation. But given that we see both dark and light alleles in Africa for a long time, I would argue that there could have been variation in Africa millions of years ago. And then the last thing that I was really fascinated by is the fact that the only place that we see the variants associated with dark skin color outside of Africa is in South India and Australo-Melanesia, two other places where we see darkly pigmented people. So anthropologists have speculated that dark skin color might have evolved independently in those regions due to what we call convergent evolution. But at least at these genes, we were able to show that they're on the same haplotype background as in Africa. So they were introduced during the out of Africa migration, and then they were maintained in those populations due to selection. So in summary, there's an urgent need to include more diverse people in human genomics research. We need to better understand how and why the genetic architecture of complex traits differs among ethnic groups, and how well polygenic risk scores translate across groups, and to develop better polygenic risk scores. And that's going to require integration of diverse data sets, and we need to better understand gene by gene and gene by environment impact on complex traits and on polygenic risk scores. And so with that, I will end and just thank the many people who have funded this research and the many, many collaborators we have. And with that, I am very happy to answer any questions that you might have.
Thank you very much, Sarah. That was a brilliant tour of all the genetics that you have done in so many diverse populations. I'm going to take the, everyone's encouraged to use the Q&A button in Zoom, or if you like, you can use chat, but please use Q&A if possible. Just type your questions in there. And I have a lot of questions, and I'm going to keep asking them till someone else raises their hand.
For now, I'm leaving the slides up just in case somebody wants me to like refer to something.
Great,
but,
great,
okay.
great. So very first question, this is really putting my National Precision Medicine hat on. I think it's incredible that you have dedicated your career to doing something very difficult, which is the easiest thing. Actually, it's not easy, but it's easier, is to stay wherever your institute is and do your genetics over there. It's just the path of least resistance. What made you do the, crazy and much harder thing of engaging with communities across the world, and really saying it's kind of a crazy idea, right? Leaving your comfort zone and saying, I'm just going to make a long-term bet and a long-term investment that takes a huge amount of upfront effort before you start seeing rewards much later.
Well, that's a great question. And so first, to give you an idea how crazy it was, when I started this, I was a faculty member at University of Maryland. I had no experience doing field work. I didn't, I actually have an anthropology degree. So part of the answer is, when I was an undergraduate, I had an anthropology and a genetics degree. And I've always had a passion for studying human evolution. And frankly, I always wanted to do field work. But when I was at Yale in the medical school doing my my PhD research, there's no way my advisor was letting me out of the lab. So it was my passion. I couldn't wait to go to Africa. I was so excited and I love it. I mean, for certain people, I mean, I don't know, for people like me and for others in my lab and our collaborators, we love doing that work. But when I first went out there, I had no idea what I was doing, never did field work, didn't know anything, went by myself, okay? All by myself out there, led a research team. And we basically practiced on ourselves in the lab, and we just tried to practice the different methods. And at that time, we didn't have a generator, but I figured that we're not going to have electricity, so I custom ordered a centrifuge that could get hooked into the car battery. And we just kind of learned as we went along. And I'm always frankly amazed that this actually worked as well as it did. But to me, it's not, you have to have a passion. I mean, that's it. And basically you have to be genuinely passionate and interested in this. And it's just because it's sort of the area that was the least explored, I feel like we're constantly discovering completely new things. And it's not that we have to go to Africa to do this, right? There are plenty of populations in Asia and other places of the world where you can also discover very new things.
Okay, so passion matters.
Yes.
Thank you. I have lots more questions, but out of politeness, I'll defer to the people who typed in the Q&A. First is from Swain Chen. Thanks for the great talk. Can you go back to the Bayesian skyline plot for the effective population size over time? So the main question is the strong correlation you see in different populations over the past 5 to 10,000 years.
Oh, yeah. That's an easy one to answer. You know why? Because don't believe any of this. So basically, this approach doesn't, I should have said it when I actually presented this, but it does terribly for making inferences about recent changes in effective population size. So the only thing we really can say with more confidence are these older events. Now, since this time, we've actually applied there are other approaches you can apply to look at more recent events. And for example, when we looked at, when we did that, we found, I don't even see where they are here, but because I haven't labeled them all, probably the Hadza are somewhere around here, but we found that they went through this really dramatic population crash, like in the last 500 years or so. The census of that population is only 1,000. So you can get better estimates, but just for this purpose, ignore those. So I'm sorry, I should have said those, it doesn't do well at recent times in the past.
Got it. And I guess at times far in the past, on the right-hand side of your plot, it's the model essentially says that we were all one population.
That's right. That was what was also exciting, is that you see this divergence starting at around 250,000 to 300,000 years ago, which is right when we see the fossils of anatomically modern humans. And that's when we start seeing divergence between populations. So that was pretty exciting.
Fantastic. All right. Next question from Volker Patzel. Some 8 to 9 million years ago, jawbones found in northern Greece in the 90s led some people to suggest that human ancestors evolved in Europe, not in Africa. Your comments?
They're wrong.
Okay.
I mean, that's true. So plenty of people have proposed that, but I think if you were to ask, and I'm not a paleobiologist, so I just know what I read, but from what I've heard, there's a very strong consensus that modern humans arose in Africa, but the place where there was a big surprise, let me just go back here. Hold on 1 minute. Let me find. The surprise was, so there was a debate between people who thought that there was a multi-regional origin of modern humans, so that they arose in Asia and they arose in Europe and they arose in Africa simultaneously. And one of the reasons that they argued that actually had to do with the shape of the incisors in Eastern Asians, was very similar to some of the more ancient fossils that they had found of Homo erectus from that were over 1 million years old in Asia. So some of the paleobiologists had argued, no, it had to have there had to be independent evolution because we see these shared characteristics for some traits. But the problem with that model is that in order for that to happen, if you're going to have, modern humans evolving all over the world, you have to have a lot of gene flow. You have to have a lot of migration and admixture. And all of the estimates of the size of the populations was that they were really, really small. And so it would be really hard to have that level of gene flow across populations. So in the end, and the more molecular data we got, the more clear it was that all modern humans arose in Africa. But then it was only after Svante Pääbo's group and others had sequenced Neanderthals, and then they sequenced the pinky bone from something, found a whole new species by looking at the DNA. They didn't actually do it from the fossil, and they called it Denisovans because they found it in the Denisovan cave in Siberia, actually. And now we know that the out of Africa model is not 100% true because there was interbreeding with these groups. And actually Denisovans, we see, apparently they see, people who look at this, see a lot more of the Denisovan ancestry, for example, in Australo-Melanesia, in East Asia. So there is some truth that we see these very old lineages now in some populations today, but it's a relatively small proportion of the genome.
Thank you, Sarah. Quick comment from Swain Chen again, saying he misread the x-axis and he was asking about the convergence in population size hundreds of thousands of years ago, which you already
yeah, which I already mentioned. Yes, that's right. Okay, thank you.
And then this question from CC Core in GIS. Hi, Sarah, given the lovely G6PD study you published in 2001 about malaria resistance, what do you think about the possible mechanism now that the recent malaria gen data suggests that G6PD deficiency protects from cerebral malaria, but is associated with the increased risk of severe malarial anemia, so kind of opposing effects in some sense?
That's interesting. Yeah, I mean, I guess what it's telling us is it's quite complicated. And another example where, in this case, it's on the X chromosome. So you're going to have, hemizygotes. So it's even more complicated that there's different selective forces potentially in men who are hemizygotes and women who are heterozygotes. But again, it's just those balancing. That's probably why it's not at even higher frequency, right? It would probably be at higher frequency if it didn't have some negative effects as well. But that's a really interesting observation, that because this had been speculated for so many years, and it's only recently when they have these big data sets that they could actually show that.
Thank you, Sarah. Try to quickly go through all the other questions. From Kanika Jen, do you think it'll be useful to genotype a few Europeans as well to look for additional genes in skin tone?
Oh yeah, well, there are plenty of people doing that. So the UK Biobank project, for example, they're sequencing now, they are starting to sequence many people. There are many other projects going on. But in terms of looking for correlation with skin color, I think, in the UK Biobank, they have very broad categories like they ask people, are you really light? Are you kind of light? Are you kind of reddish? Do you tan easily? So they're actually finding information, but if you really want good information, then you've got to actually measure the, melanin levels. But the big mystery is Asia. So that's where nobody's been able to find. Almost nothing has been found in Asia. And that's a really big mystery because it looks like an example of convergent evolution in East Asia of lighter skin color. And people just have not been able to find the variants. And that could be, again, because of this European bias, that there just hasn't been enough done in Asia.
Thank you, Sarah. That gives us something exciting to chew on in our Asian precision medicine programs. From JJ Liu, genetically, lineages are generally correlated with linguistic lineages, but not perfectly. So can you comment on the divergences between the two?
All right, let's go to that one. Where did I put it? Down here. That is true. Trying to think of a couple of the examples. Well, what started my entire career was I was working in South Africa. I did a short postdoc at University of the Witwatersrand, and I attended a meeting that was on the history of the Khoisan, the click speaking groups. And I just was fascinated. They had historians, archaeologists, linguists, geneticists, and people from the populations. And I just thought this was like the coolest thing. And I wanted to know, where did these groups come from? And in fact, I had asked the people there, what's the most interesting thing I could look at? And they said, go study the Hadza and the Sandawe who live in Tanzania and are classified as the same language family. We still haven't figured it out. Like 20 years later, more than 20 years later, I still don't know. It's a very complex history, but it does not look like there's a super recent, we think there's actually been probably some gene flow between them in the past like 10 to 20,000 years. But that's an example where speaking with clicks could have been the ancestral state actually, and that other populations lost the clicks. That's one possibility, including pygmies who no longer speak their indigenous or traditional language. They speak the languages of whatever the neighboring groups are. But there were some others, and there's more we have in another paper, but I think the Mata, for example, if I remember, these guys, I think we sampled them in Cameroon, but they're actually people who had migrated from East Asia, and I'm forgetting, but those were the kinds of things where we'd see things that don't match. Also, where was the other one? Let's see here. Yeah, I just can't think of any other quick examples, but we do see that. And sometimes it's either because there's been a language switch, borrowing, exchange, or sometimes gene flow will alter what the genetic ancestry looks like.
Okay, fascinating mysteries there. And I think we'll see this all over the world, but it's fantastic that you've cataloged this and characterized this in such a diverse population. One more question from Swain Chen. The first gene you described for skin pigmentation was SLC24A5.
A5.
Yeah, had separate gene mutations and enhancer mutations. And you mentioned that the genic variants were localized to Africa, but the enhancer variants were polymorphic and out of Africa. Is that right? Or was there a convergent evolution in the enhancer separate from the variants in the gene body?
So I think we're talking about different things, because at SLC24A5, that was a non-synonymous variant. And it's the same one. It actually was found initially in zebrafish, I think, that they had found this, and you could see through the fish, so you could see very easily the change in the color. And then when they found this, they collaborated with someone who was had a data set of European and maybe African-American skin color, and then we're able to show it's correlated. But that's a non-synonymous variant. So I think what you're referring to is probably the regulatory variants. And I guess what I should point out is that these are exactly the same allele. They have the same origin. So actually the allele associated with light skin is ancestral, not the dark skin. Light skin was ancestral in Africa. And then it was introduced out of Africa during the migration. So it's the same enhancer, the same impact on gene expression in the Africans and in the non-Africans. So I don't know if that answers the question.
Yeah, I'm guessing it was this gene then, because this has an enhancer variant as well.
Right.
From Suma Tiruvai Pati, Thanks for interesting talk. It is said that India was part of Africa, which split millions of years ago. Could this be the reason for the dark pigmentations shared by some groups in Africa and South India and the other region towards Australia? Well, I know the answer, but I'll let you say it.
Well, yeah, so that's too long ago, because millions of years ago would have predated, that would have been, at the time of our common ancestor in Africa. So that can explain it. But what some people have hypothesized, and you know what? Now I'm seeing they don't really even show it in this figure so much, but there's a big debate. So this debate is, was there one migration out of Africa as shown here, and maybe an early, once they migrated, then one group went south, into Australo-Melanesia, South India, or were there two migrations? So some people have speculated that there was an earlier migration through the Horn, through Saudi Arabian region, and then into, some have even settled on the coast. Like some people have even speculated, did they have boats or something? Maybe. And did they leave Africa and kind of stick to the southern route and go to South India and then over into Australo-Melanesia? This is a huge debate that continues today. I personally don't think anybody has sufficiently answered this, because if you look at some markers like mitochondrial DNA, if I'm remembering correctly, will show some connections. Certainly you'll see some between like South India and Melanesians and maybe in Africa. People looked at the whole genome. Some people argued, they came to opposite conclusions. So one group concluded that there were two migrations and the other group said, no, there's only a single migration. But the problem is that even, let's say there were two migrations, it was such a long time ago, and there's been so much more recent admixture that it's very hard to reconstruct that small amount of sort of the indigenous. But that's why I thought it was fascinating when we saw that these alleles associated with dark skin are not just in Africa, but they were introduced into South Asia and Australo-Melanesia, would certainly be consistent with the southern migration route and could perhaps help to explain some of the phenotypes that are similar between populations, like in the Philippines with hair type and skin color and things like that.
That's tantalizing. How old are people saying that southern migration was?
About 80,000 years. So people were saying that would be on the order of 80,000, and the other would have been 60,000, like 50 to 60,000, something like that.
Okay, so still well after the emergence of modern humans.
Oh yeah, definitely after the emergence of modern humans. Yeah.
Okay, got it. I know we're keeping you late, Sarah.
No, so that's okay. Don't worry.
But okay, let's say one last question. Don't want to be rude to you. One last question from Swain. Although we could go on for hours. So from Swain again. So yeah, it was MFSD 12, but the genic alleles, if expected to be on the same haplotype, seem to be strictly localized to Africa, while the enhancer mutations were found in Asia and Papua New Guinea and so on. So can you talk about whether there are different histories?
Yeah, I see. Okay, let's find it. Where to go? There. Okay, so now I see what you're saying. Yeah, so that is correct. Sorry, these arose independently. These are totally independent associations, and they show different evolutionary histories. Okay, so this is a gene that others now have shown that MFSD12 is playing an important role in the pigmentation in, I think, dogs and horses, and in Asia. Actually, they've shown in Asians as well as in Native Americans. So now it's been shown this is playing generally a really important role in pigmentation. And it appears that there's this regulatory variant that's influencing expression, and then there are these variants within the gene, and we don't know yet exactly what they're doing. There is some evidence we think that they may also be playing a role in gene regulation or possibly alternative splicing. And just based on the history, it happens to be that these are very specific to Africa, whereas the, excuse me, the other variant was introduced during this migration out of Africa. So different evolutionary histories. And it could sometimes just be chance, or it could be possibly even the impact on phenotype. Could be stronger, I forget, it might be with this one, and for some reason that was maintained and the other one wasn't.
Got it. Okay, I'm going to call this to a close with one last comment. I kind of, as a, to, you know, how can researchers follow your example? And just to comment, either follow your passion, like you suggested, or if you want a cold, calculated reason, then that would be that find something really important that is yet no one is doing.
Right.
And if you can find something like that, then it's worth investing in, and you know, it doesn't matter how much you need to invest in it, it'll be worth it.
That's what I think. And it can be, it's not for people who are impatient. I'll tell you that.
Exactly.
And the other lesson is you've got to do it the right way. So if we had rushed this, like a lot of people were doing when we started, and they would do what's called helicopter genetics, and they fly in and they would take stuff from markets, like just take people's DNA, didn't get research permits, didn't go through ethical review. And that caused a lot of problems later for these people and for people in the field generally. So I think what was important was to make sure right from the beginning, get all the permits, get all the ethical reviews, do it the right way, because if you do it the right way, you're going to be doing it for the next 20 years or so of your career. So that's the other lesson. Just take your time, do it the right way right from the start.
Great note to end on, Sarah. Thank you once again. It's been a privilege.
Oh, thank you. It's been my pleasure. Take care, everybody. Bye-bye.
Bye-bye.